Hello and welcome to your second Ed Puzzle for AP Environmental Science. We are continuing into the wonderful world of Chapter 1, but we're actually be focusing on the scientific method and experimental design as a whole. You guys will actually be designing your own experiments very soon, and design of an experiment is a huge part of our AP exam. Well, maybe huge, it's like 12% or so. So I want to make sure that you guys feel very familiar with this. We're looking at the scientific method as a whole. Some of this may be review for you guys, and some of this may be completely new. No matter where you are on the spectrum, hang in there. Take some good notes for sure. That way you can use it on your quizzes. But when we're looking at the scientific method and we're looking at that process of developing an experiment, a lot of times people will think that you start with the hypothesis, but I want to make sure that you guys know that before we even create a hypothesis or a prediction that we're going to experiment, and test, we want to start with observations, right? What's going on in the world around us? What do we already know about a topic? And then from there, we can make a prediction. We can collect data to see if we support our prediction. We can see if our results are acceptable for the hypothesis, or if they do not follow along with our hypothesis. And if it's accepted, we can actually disseminate that. So we're going to be looking at each of these in depth. I'm going to start out with that making observations. Again, this should precede making a hypothesis. And when we're looking at observations, we want to make sure that these are objective facts, not inferences or predictions. I don't want to bring in feelings or suppositions on this. I want them to be provable. I want them to be definitive facts. So let me show you guys what I mean by that. So for example, this is an objectable observation. There was an axe next to the bloody dead body. Good morning. We're just watching this in the morning. That is an objective fact, right? There's no beating around the bush. There is an axe and it's next to the bloody dead body. An inference, hypothesis, prediction from that could be the axe was the murder weapon. There is no way for me to prove that. And I guess we could through the fact, but this is an inference, right? It's not a fact. Somebody could have been killed another way, and then like a lumberjack walked by them and dropped off an axe next to the bloody dead body, right? This is an inference. Another objectable observation, the axe of the crime scene was found 10 meters to the left of the body. There's no beating around the bush with that, right? That is an objective fact. An inference from that could be the killer was left-handed and threw the axe to the left. We don't know that to be 100% true, but we are inferring it based on the observations around us. Also, when we're getting into creating a hypothesis, we also want to take into consideration, what do we already know? What is our background information on this topic? How can we further the scientific field based on what we already know? So if I know for sure that rocks can't float, I'm not going to design an experiment, can rocks float? I already know that they can. So I'm not going to waste my time or other people's time designing an experiment that already tests something that I know. So we know some of the observations of the world around us. We know the background information. Now we can create a hypothesis, and we're going to spend at least half a day creating hypotheses, because I feel like this is where a lot of times people struggle, because some key parts of hypotheses is we want them to be testable. We want them to take a particular stance either way. And we want to be able to actually experiment on them. But it also contains prediction based on our observations. So here's some examples of some bad hypotheses. And we're going to break down what is bad with these and how we can actually improve them. So I'm not going to read all of these, but you can take a second to read these. But a lot of these are bad, especially chocolate may or may not cause pimples, or I think salt and soil causes plants to fertility. Why are these bad? Well, let's think about what we want our hypotheses to be. We want our hypotheses to be statements, not questions. You want to keep away from question marks. I know a lot of times in middle school, they tell you that I think, or I predict, things like that. But we really want these to be objective statements that can be tested in one direction or another. So don't put I think, don't put I predict. We want our hypothesis to say this causes this, or so like X causes Y because of B. We want them to be objectable statements. We want our hypothesis to be testable. I'm going to go back to this last slide. This last point is a bad hypothesis. Sparrows are happier than birds because it's not testable. If I don't have a way to measure happiness in birds, then this experiment is not testable. We need it to be something that's objectable, that's something that is measurable. All right, so we want it to be testable. Finally, we want our hypotheses to take a specific stand either way. I don't want to say, for example, chocolate causes pimples. I want to say chocolate causes more pimples than because. So I want my hypotheses to create a stance and also say why we have that stance. So here's those same, oops. Oh, sorry, I almost forgot. I guess this hypothesis is actually not too bad. It is an objective statement. It is creating a prediction. It is testable because you can mix them, but kind of funny. Okay, so this is the same slide as earlier, but these are better versions of that. So chocolate causes more pimples than vegetables. All right, we're making a statement, we're giving it a direction, and we're taking a stance. There will be a greater growth of pimples with chocolate diet than with vegetable diet. Salt and soil causes plants to grow slowly. I'm just going to take out that bottom one because, again, how do you measure happiness? Maybe you can say greater time spent flying. That could be a measurable thing because we can measure how long they're in the air, but happiness in birds is, as far as I know, we don't have a test for that. So, all right, null hypothesis. A lot of times students will be confused about what exactly a null hypothesis is. So earlier, these are what we call experimental hypotheses. It is what we are predicting, what we are experimenting on to see if there is a trend in our data. Sorry, I went the wrong way. Null hypothesis is not the opposite. So sometimes people will say, let's say if your experimental hypothesis is chocolate causes more pimples than vegetables. Sometimes people will think, oh, the null hypothesis is just the reverse. Vegetables cause more pimples than chocolate. But the null hypothesis, null means no. So a null hypothesis says that there is no relationship. So for the chocolate versus vegetables causing pimples, the null hypothesis could be diet does not have an effect on pimple growth. It's essentially taking my two variables, diet and pimple growth, and saying, no, there's not actually a relationship between those two. So we have a null hypothesis in case you design your experiment, you run your experiment, and you're like, crap, cola and cheese, there is like, this does not support my hypothesis. Then you can fail to accept your experimental hypothesis and accept your null hypothesis that perhaps there is no relationship between these two variables. All right, we made some observations. We created a hypothesis. Now we want to actually design my experiment. Now there's a couple things that you guys will be working through as a class on how to actually design an experiment. Usually you're going to have two different groups or two different types of groups. Not always, but most experiments are going to have a control group. These are going to be your groups or your individual seeds, your sample that's not actually receiving or accepting any treatment, right? So if I'm seeing how does salt in soil affect plant growth, my control group is going to be plants that don't have salt in the soil that I can then compare to the plants that do have salt in the soil, my experimental groups that have that independent variable attached to them. So it's kind of a baseline to see, was there effect or was there not effect when I put salt in the soil as compared to no salt in the soil? Sorry, another funny picture. Well, I guess we're the control group. Okay, other things that you should be familiar with is our variables. A lot of times people will get independent and dependent variable mixed up, but it is extremely crucial that you do understand the difference between the two. My independent variable is going to be my treatment. What am I actually changing? So diet, the different diets that I'm giving, that is different treatments for the pimple experiment, or for thinking about the salt in the soil experiment, the soil type, whether it has salt or no salt, that treatment that each individual plant is receiving is the independent variable. Your dependent variable is dependent on the treatment or independent variable it is receiving. It's the results, what actually comes about due to the independent variable. So plant height, the number of pimples on face, the measurable amount that results from the independent variable is our dependent variable. And of course, we'll always want to have constants. What is consistent between the groups? So we want to make sure, we're only testing, or the only thing that's different is my experimental independent variable. Everything else, temperature, moisturizer, amounts of food eaten, everything else is consistent. The only thing that's different is that independent variable. That way we can isolate any results as being attributed to that independent variable. A good way to remember independent versus dependent variable, or a way, I don't know if it's good or not, is mixed versus dry. My manipulated independent variable, it's always on the x-axis, this is our treatments, then results in a dependent variable, which is our responses, our results, and they're always graphed on the y-axis of our graphs. All right, some common issues with experimental design. Hopefully we won't run into these, but I mean, with the nature of us having just one class, we'll see what we can do. But in the field of science, if your sample size is too small, it's hard to actually generalize your results to the world around you. If you had like only 2 things being tested, you can't make a blanket statement that this is the truth for everything involved if your sample size is really small. You also want to repeat. In an ideal world, you would want to repeat to see if you are able to get the same results. If you don't get the same results, even when you repeat, maybe something was just a fluke. You want to make sure that you have a control variable. That way, you know, is my results attributed to that experimental independent variable? And if you have too many independent variables going on, like if I have differences in soil and I have differences in light and I have differences in what water I'm actually giving to those plants, and I see differences, cool, yay differences, but how will I know which actual independent variable caused those differences? I want to isolate it, have only one singular difference between my groups. Otherwise, I won't know which one actually caused a change if I see a change in my results. Okay, so let's try it out. Let's practice, practice, practice. I've got an experiment. Let's see if we can try to figure out all these different parts. So Lois wants to find out whether feeding babies mashed carrots improves their vision. She borrows 50 babies casually from her son Stewie's daycare. 25 of them are giving a normal diet. 25 of them are given mashed carrots as their evening meal every night. She gives them a vision test. What is my independent variable? What is being changed or what is different between my groups of babies? That's going to be the diet of the babies, right? Carrots versus no carrots. That's the only difference that is between these groups of 25 babies. The dependent variable, what results and what actually comes about, what am I measuring as a kind of a result of the independent variable? That's going to be the vision. So that's going to be my measurable results of the experiment. My control group, it's going to be the ones that aren't getting carrots in their diet. They're just getting their normal baby food. So we're comparing them to my experimental group that has that independent variable added on, that treatment, diet with the carrots, that's the treatment we're experimenting on. My constants, what is going to be the same between the two babies? Maybe their morning food, maybe the age of the babies, maybe the overall stimulation around the room. Age, sex, baseline vision, amount of sleep. We want to make sure it's the same test. So everything that's consistent between the experimental and the control group, those are going to be our constants. Okay, let's keep going. We've done our experiment. Now we're going to look at our data and try to figure out what does our experiment tell us. We're going to take 2 different types of data. Oftentimes, our qualitative is going to be the quality. Oftentimes, this is going to be things that we can visually see. So color, smell, texture, anything that I can see, something that's descriptive is going to be my qualitative data, which is different from my quantitative, like think quantity data, numerical data. So am I measuring in a quantitative way, number, length, weight, amount? Those are all going to be our quantitative data. Once we've collected our data, then we can actually go on and interpret it. Some just general terms that you should be familiar with in the land of experiments is replication. So the number of times that I do the experiment over and over again, hoping to get the same results every time. That way I know that I'm precise and I'm accurate. Sample size is the number of individual data points. So if I am testing 6 tomato seeds, my sample size is 6. So the number of times that individuals are receiving that treatment. Accuracy and precision, I'm going to have a slide on next, because these are 2 things that get kind of mixed up. We want both. Accuracy is how close what I actually obtain is to what I want. So how accurate is my data? Precise is if I'm doing the experiment over and over again, am I getting the same results every time when I go through those different replications? How close are all of my measurements? And uncertainty is how much that measure differs from the true value. How good was my experiment at measuring what it's set out to measure? Or do we have a lot of degrees of freedom and uncertainty in my data? So a good way to visualize and try to differentiate between accuracy and precision, we can think of a dartboard. This one on the left, not very accurate throws, right? If I'm trying to get to the bullseye, these darts are like way out of there. They're not where they're supposed to be, but they were very precise. Every time I threw those darts, they all kind of got in the same area. So this is very precise, even if it's not very accurate. In this one, I have greater accuracy. I'm a lot closer to what I want to get that bullseye, but I'm not very precise. Every time I throw that dart, it's not where I need it to be. It's not in that bullseye. Ideally, our experiments are going to have high accuracy. They're going to measure what they set out to measure, and they're also going to be very precise. When I repeat this experiment, I tend to get the same thing every time. Okay, test yourself. I want you guys to take 10 seconds or so, pause the video, look at these. What are these? Vocab words on the left. We got some definitions on the right. Try to match the vocab words with each of the definitions. Pause the video. Okay, we're back. So now that we're back, let's see how you did. Woohoo! Answers, answers, answers. So if you didn't get all of these right, that's okay. Learn it. Make sure you've got it down for our quiz, which is coming up pretty soon. Okay. I thought it was global warming. Okay, we're almost done. Once I've collected my data, now I want to present my data, present my results. It's going to depend on what kind of data you have, on what kind of graph that we're going to be selecting for presenting my results. I want to make sure that I've got what is my independent variable, what's my dependent variable. My independent variable is always going to be on the x-axis. If it's a graph, like if it's a line graph or it's a bar graph, pie charts, I guess, don't have x-axes, but anyways, we want to know also if my data has a category or if it's continuous. That's going to affect what kind of graph that we're going to use for our data and what graph is appropriate for the data. So our categories is whenever I have no in between. So if the results I'm receiving is like yes, no, or if I'm giving people a survey and the survey has options such as strongly disagree, disagree, no opinion, agree, strongly agree, those are categories. There is no in between each of those. And so because of that, my data is categorical. Again, we're looking at the dependent variable to figure out what kind of graph I'm going to be using. So gender, color, rank, species, brands, grades, these are all categorical things. Usually they're going to be represented in a pie chart if it's parts of a whole. So kind of like with that strongly agree to strongly disagree. This is a really weird pie chart, but anyways, a pie chart could be good for representing that the strongly agree to strongly disagree because those are categorical. Also categorical data could be bar graphs. So if somebody asks, how in the world did you understand or how did you get this presumption? And they answer, they've got one of 5 options. This would be categorical. A bar graph would be really good for this. Bar graphs are also helpful if you're looking at averages. So if you're looking at the average between data sets and you're comparing 2 different groups based on their averages, that could also be a good one. But I'll tell you guys if your group needs that kind of data. Continuous is whenever I'm looking at my dependent variable and it's a longer range. So for human height, height is a continuous data point, right? You're not either tall or short, right? You're like, you've got 4 foot to 7 foot and everybody is somewhere in between there. Generally speaking, if you're over the age of 15 or so, but you know, it depends. It depends. So this is whenever we're along a range, along a spectrum. If you're looking at different intervals, so if you're looking at age or height, but you're dividing it in by inches and you're seeing frequency or how many people for each of those, this would be a histogram. Histogram we use a lot for populations. You're not going to really use it for your tomato plants for our experiment because you're not going to have hundreds of data points, right? Our data points is going to be like 12. So because of that, we're not going to do a histogram. But when you've got a lot of data, histograms are really good for that. If you're looking at cause and effect, if we're looking at trends over time, a line graph could be really good for that. Line graph might actually be really good for y'all's data because we're taking a lot of data points over time. Maybe we could do average height, but we'll figure that out. If you're looking at the relationship between two different variables and we're seeing if there's a correlation between them, a scatter plot would be really good for that. Scatter plots I would not recommend for the tomato lab, but it's good for other kinds of experiments. Okay, we're almost done. We're on the last step. Yay! Analyze data, draw conclusions. So once we've actually taken our data, we're looking at the graphs, now we can interpret our results and say, do we support the hypothesis or do we not support the hypothesis? Do we support the null hypothesis? Was there not a relationship at all? When we're going between these, we're going to really look at inductive reasoning versus deductive reasoning. I get these two confused all the dang time. I'm going to be completely transparent with you, but this is kind of how I have coped with understanding it and how I've gotten this far with it. Inductive reasoning is whenever I'm making a general statement based on one individual. So we're starting very small and we're generalizing to the whole population. So personal example, I have a 3-year-old cousin named Chris. Chris screams all the time. So if I saw specific example, Chris, 3-year-old screams all the time, and I made a general statement, all 3-year-olds must scream all the time, that is inductive reasoning. We're starting small and we're making a general statement. Deductive reasoning is the opposite. I'm taking a general statement and applying it to a specific example to try to make an inference. So let's say I know that air pollution kills trees, okay? And that's a general statement, air pollution kills trees. And then I'm driving along and I see a dead tree. A deductive reasoning could say, let me take that general statement, air pollution kills trees. I see that individual dead tree and I say, that tree must have been killed by air pollution. That's deductive reasoning. General statement, applying it to a specific example. We'll go over that more in class, I promise. Okay. Finally, we're going to write our conclusion. We want to restate the problem. We want to say if our hypothesis was supported or not, why was it supported or not, what data supports or goes against our hypothesis. We really want to get into what's going on between our independent variables and dependent variables, the relationship or lack of relationship that we see between those, and finally, any trends or lack of trends that we see in our data. This one's going to be a big one for us. Causes of error. Humans are imperfect. Goodness gracious, no, we make mistakes all the time. Your experiments are probably going to have mistakes in it. There is never in the history of any science been an experiment that has no errors. Possible causes of errors, you got to think about it because that's when you take into consideration, yes, this is what I see, but here's what possibly is influencing what I am or am not seeing in my data. We want to actually apply this to the real world. We want to connect it back up to that top level, that background knowledge. And finally, we want to have that final summary statement. We are almost done. I'll remind you guys when we get to this part of our actual experiments, never use I, we, us, me, no pronouns. We want to make sure that it is very objective. So the experiment, the scientist, or it was found, not we found. We want to make it as objective as possible. Ideally, then you would then publish your findings. Unless you make some groundbreaking discoveries in your tomato seeds, we're probably not going to get to this part, but usually then we publish our results to the scientific community to help further the overall understanding of the field of science. If then this experiment goes to the big leagues and it ends up actually being tested over and over and over again, everyone's finding the exact same thing, your hypothesis could become a theory. For example, the theory of gravity, right? I generally know, and all the gravitational experiments show that if I take a rock and I drop it off my balcony, the rock is going to fall. That supports the theory of gravity. As far as I know, there's very few counterexamples that disprove gravity. So that's why it's graduated from the hypothesis of gravity to the theory of gravity. All right. Again, this is just reiterating everything that we've just gone over. So yay, look at it if you want, but I feel like we've gone over it quite a bit. You're probably tired. Some problems and difficulties with having experiments in environmental science. I'm not going to read all these because these are gone over in your text, but the main one is that there is no control planet Earth to compare with. We're looking at what we see in the world around us as human beings affect and alter our planet, but we have no baseline to compare it to. I guess you can think of like different regions, like maybe if some regions are experiencing more air pollution than others, you can compare them, but it is kind of difficult because there is no baseline, right? Anyways, we keep going. There's a lot of interacting parts, the atmosphere and the ocean and our grasslands and our plants and the humans, everyone's kind of mixing in together. So it's hard to isolate one independent variable saying like, this is causing this, because it could be a number of things that are affecting different parts, different factors. We're going to end today with a riddle. I want you guys to try to meditate and see if you can figure it out. What English word has 3 consecutive double letters? Think about it. Let me know what you got in class. We'll reveal the answer very soon. I hope you guys have a fantastic day. You made it through a very long ed puzzle, but I hope you guys have a great day. Let me know if you have any questions, and I will see you all in class.